Edvotech Instructional Videos presents Pre-Lab Preparations for Exploring Biotechnology with a Green Fluorescent Protein. Preparation for Module 2, Isolation of GFP. For this prep, you will need TEG buffer and lysozyme to create the lysis buffer. Each group will need 2 plates of LB agar containing ampicillin and IPTG, 2 inoculating loops, sterile 1 ml pipettes, and 1 microcentrifuge tube, and finally, control GFP plates can be provided if needed. Step 1. Add the entire contents of the lysozyme to the bottle of TEG buffer and swirl to mix. Label this solution as lysis buffer. Step 2. Dispense 500 microliters of the lysis buffer for each group. Step 3. Assemble the remaining materials for each student group. Step 4. If needed, retrieve the 2 control GFP plates that you prepared earlier. These can be used by students who may have had poor transformation results. You are now ready for students to perform Module 2, Isolation of GFP.